So our featured speaker today is Hamutahl Cohen, Extension Advisor with the University of California, Agriculture and Natural Resources, UCANR. She's based in Ventura County. As an entomologist, her primary focus is on integrated pest management of insect pests in agriculture, including identification and monitoring, developing cultural and biological control methods, and evaluating new chemical control products. She works on all crop types grown in Ventura County, including avocado, citrus, strawberries, cole crops, and greenhouse ornamentals. Hamutahl received her PhD in environmental sciences and previously worked in extension at the University of Florida. She has over 13 years of experience in entomology, horticulture, and ecology. She's heavily involved in communication and research, and she has authored books, articles, and blogs for academic, industry, and public audiences, and has given numerous workshops about topics such as climate change, sustainable food systems, pollinator conservation, pesticide safety, and more. This is now the second time this year that I'm here inviting a featured speaker from the agriculture sector to come share their IPM work with our group. And as you all know, here in San Francisco, we're typically focused on urban IPM. We don't produce agriculture in the city and county of San Francisco. Hamutahl works on sustainable pest management methods for fruits and vegetables, and we do all need to eat, and the way that the food that we fuel our bodies with is grown really matters. California grows a significant amount of the produce that's distributed around the country, so our state is in a position of power to lead by example when it comes to sustainable pest management and prevention in production agriculture. So today, we will get to hear Hamutahl discuss the IPM triangle, cultural, biological, and chemical control in Ventura County agriculture, pesticide resistance, and the applicability of lessons learned in agriculture to urban environments. So I'm ready to pass it on over to you, Hamutahl.
Wow, that was a great intro. Before I start, are you seeing my slide and everything?
We're seeing your slide. Your webcam is off.
Is that okay?
Yes, that is fine. Just wanted to make sure it was intentional.
Yeah, that way we can just focus on the presentation. Thank you for that introduction. You covered really what I wanted to share about myself, but I did want to mention what UC Cooperative Extension is for folks who are not familiar. We're basically an arm of the University of California. We're a network of researchers and educators charged with meeting the needs of our local community. So in my role, I work as an entomology advisor in Ventura County, and I work in lots of different crops that we have: strawberry, brassicas, citrus, avocado. And in addition to conducting research, part of my role is also sharing out what studies are being done and the results that we're finding with local growers, with pest control advisors, and with other industry members. So I do have this understanding that most of you in the audience don't work with field crop agriculture necessarily, but you have these IPM backgrounds that you apply to urban systems. I thought I could share a few of my projects today in the context of IPM and the IPM triangle, and then we could have a discussion afterwards maybe about how field agriculture differs from urban agriculture or urban horticultural systems when it comes to implementing IPM. So quick background on IPM, in case anybody doesn't know, IPM basically refers to using a suite of tools to accomplish pest management. And the basis of an IPM program is identification and monitoring, which is accomplished through scouting, note-taking, and identification practices. The next rung of the ladder in an IPM program is cultural control, and that refers to cultivation practices that reduced pest populations. An example that I really like for cultural control is crop rotation, where you alternate what crops you grow in a field so that you break up target pest population. After that comes mechanical control, and that's when you use physical methods to manage pests. So that might be traps, barriers, or hand picking. And then there's biological control, which is the use of naturally occurring enemies to control pests. And then at the top of the triangle is the tool that you reach for last: chemical controls, either organic or conventional. I currently have projects running that fall within this IPM triangle. So I was going to give you 4 projects that I'm working on. And as we talk about those projects, we'll talk about these different types of IPM practices. Okay, so the first project I'm going to talk to you about falls within the realm of cultural control. So again, that's manipulations of the ecosystem that you're working in that makes it less friendly to the establishment and to the proliferation of pest populations. So it could be a lot of different practices. It could be selecting the right cultivar that's maybe pest resistant. So if you're working with grass, picking grass cultivars that are more resistant to pathogens. It can be weed management if your weeds are harboring pests. It can be sanitation. So if you're pruning out a tree that's been infested with pests, making sure that you dispose of those prunings so that the insects can't escape. Rotating crops, changing irrigation practices. Most of my examples that I give have to do with insect pests, but these practices are applicable also to plant pathogens and weed pests as well. So the first project I'm going to share is an example of cultural control. This is work with my colleague Emma Volk, who's a greenhouse advisor, and it's with cut flowers. Cut flowers account for approximately 600 acres of Ventura County agricultural land. And the most popular cut flower that we have here are gerberas. These are like the grocery store flowers that you pick up at like Ralph's or Vaughn's. As an ornamental, because they're valued solely for their aesthetic appeal, our growers have a very low tolerance for pest damage. But they do indeed have a lot of pests. And one of the biggest pests that they struggle with is Western flower thrips. So thrips are insects in the order Thysanoptera. Thysanos means fringed and ptera means wing. They have these feathery wings that are kind of like narrow. Western flower thrips are kind of thrips, and they're widely distributed throughout the United States. They have lots of different plant hosts, and that includes gerberas. They cause deformations on the blooms, and those deformations basically make the flower unmarketable for retail or for wholesale. They're also capable of vectoring viral diseases like INSV, which is a really big one in our region, impatiens necrotic spot virus. And that virus causes leaf mottling and necrosis. So for control of Western flower thrips, a lot of our growers use biological control. They release predatory mites. And these mites can work really, really well, but only when the thrips populations are low. But once they get to a critical mass, growers have to use chemical controls. The way that gerberas are grown is really interesting. They're basically grown in these pots with substrates that totally lack any kind of nutrients, the way that soil does. It's just coconut coir. And growers apply essential nutrients through fertigation. However, there's been a lot of studies that have shown that if you over-fertilize a plant or you provide high nitrogen specifically, that can actually exacerbate pest population problems for pests that suck sap like thrips. And in those studies, reducing nitrogen has been associating with reducing pest levels. The challenge for gerberas is finding how much can you reduce nitrogen and still achieve pest control while maintaining yields and maintaining like a large full bloom. So in this study, we're working in an experimental greenhouse. We have 4 nitrogen treatments, and then we have groups of pots that receive one of the 4 treatments, and then we replicate it. And then we have a fertilizer that's integrated into our irrigation water using dositrons. We just started collecting data for this project this past week, but basically we are harvesting flowers twice a week, and at each harvest, we count Western flower thrips, we count any other pests that we might see, we give the flower an injury level rating, and then we measure how big it is and how wide and long the stem is as well. This is not data from this study, but I feel like I should show you some data. Since we only have one sampling date, I couldn't find anything that was interesting to show you, but I found a project from Oregon State University that was kind of similar. It looked at how thrips on flowers respond to mulch type used. And what they found was that flowers with no mulch have fewer thrips on average, likely due to reductions in nitrogen in the plant tissue. So I anticipate we might see something similar. We'll find out. The question will be how nitrogen impacts our flower harvest and thrips levels. Okay, so that's it for cultural control. Another big part of IPM is biological control. That's when you use natural enemies to control pests. And these natural enemies don't have to be other insects. They could be bacteria, for example. BT is a naturally occurring bacteria that's used for a wide range of pests. But commonly, we do think about insect predators. And the one that everyone's really familiar with is the ladybug. But I wanted to also show a picture of what it looks like as a juvenile. It almost looks like a tiny alligator. Another great natural occurring predator that maybe some people don't know about is the syrphid fly. It's a fly, but it kind of looks a little bit like a bee. It's a bee mimic, but it only has two pairs of wings, unlike bees, and has these big red eyes that sometimes touch in the middle. Sometimes you'll see it called a hoverfly because of the way it hovers as it flies. There are many different species. In my area, I know we have at least 7. And as adults, they eat pollen and nectar. They love sweet alyssum and like small white flowers. But as juveniles, they kind of look like tiny green worms. Sometimes they have this faint white stripe on the upper back. And they eat soft-bodied pests. They're extremely voracious. A single syrphid could eat hundreds of aphids or other pests in its lifetime. So this is an excellent predator that we have. We know it attacks a lot of the pests that we have in my county. For example, Asian citrus psyllid, which is a big citrus pest. Another agent of biological control are the parasitoids. So parasitoids are organisms that live in or on their host and kill them, unlike a parasite, which will feed on a host and not kill it. They're often wasps, so they could be flies, for example, and they're usually very, very tiny. So the black one here is laying an egg inside of like a worm-like pest. The yellow one is laying an egg inside of I think that's like a juvenile white fly. And then the brown one is laying an egg inside of an aphid. So those eggs will hatch into larvae, and then they'll eat the pest from the inside, turn into adult wasps, and then eat their way out. It's very similar to how aliens work in the 1979 movie Alien, if you're familiar with that movie. With aphids, you can often see when they've been parasitized. So if you look at a cluster of aphids on a plant and you see that some are puffy and have a hole in them, that means they've been parasitized. Okay. There are 3 types of biological control strategies that can be utilized in an IPM program. The first one's importation biological control, and that's useful for invasive species without local natural enemies. That's when you import natural enemies from the region that the invasive pest came from. When Asian citrus psyllid showed up in California, for example, a group of researchers from UC Riverside traveled to the region where it originated, and they found a parasitoid that attacks it. They went to Pakistan and they brought back that parasitoid from Pakistan. They did a bunch of experiments to confirm if releasing it would harm the local environment, if it was totally safe. And it was actually reared and released en masse for several years after 2011. So now it's like naturalized in California. The next form of biological control is augmentation biological control, and that's when you release natural enemies, but you're not importing in like a new species. And again, the common example is ladybugs or praying mantises. You can go to Home Depot or Lowe's and just pick them up and release them in your garden. But I wanted to highlight a couple other ones. There are commercial insectaries that also rear other insects that you maybe haven't heard of. A major pest that we have in citrus in California is called a red scale. And growers can purchase a parasitoid called a Phytis malinus and release it. They're actually quite excellent control agents if you release enough of them. And then there are like predatory beetles and other insects that you can purchase and release as well in a large-scale agricultural system. The last form of biological control is conservation biological control. And the idea here is if you're releasing natural enemies into an environment, they aren't necessarily going to stay there. And it's really costly to do so every year, every time you have a pest issue. So instead, you can promote naturally existing populations by modifying the crop landscape. We know that a lot of natural enemies rely on pollen and nectar. They either utilize it as a complementary food resource or they absolutely need it in order to reproduce. So we talked about syrphids briefly. They like sweet alyssum, which is what you're seeing in this photo with the brassicas. A lot of parasitoids like small white flowers. Things like predatory thrips and lacewings also use pollen and nectar from a lot of different sources. And there's different ways to promote these insects. You can use floral strips, or you can use hedgerows, or any other form of plantings to provide food for them year round. We can think also about conservation biological control, not just in this local context of adding flowers to a farm, but in terms of habitat loss and larger landscapes. If you look at this map, the red to green color scale shows human impact across the state of California. Scanning over this data, you can see that the areas of biggest human modification, that's the red colored areas around urban cities and around agricultural zones. And in this process of like urbanization and agricultural intensification, we've lost, I think, 90% of our wetlands, 99% of our riparian areas, and most of the native grasslands as well. In Ventura County, where I work, about 25% of the county is farmland, and the rest is just a mix of urban and natural areas. And if you're a predator or a parasitoid, this is the kind of landscape that you might live in Ventura. This is kind of what sometimes is called a mosaic landscape. So if you were to drop a pin on any farm and zoom out, you'd see that that farm is surrounded by a mix of urban, natural, and agricultural zones. So I'm currently working on a project with citrus and avocado pests to examine how habitat mosaics impact their populations and the control of them. And we have a lot of pests that impact citrus and avocado. Some are specialists, like avocado thrips. Some are generalists, like citrus thrips. They have lots of plant hosts. Some are very severe, serious pests, like Asian citrus psyllid, and some are just hanging out around at ambient low levels all the time. For a lot of these pests, we don't have a great understanding of how habitat impacts their pest levels. Let me just talk briefly a little bit more about Asian citrus psyllid, because I know I'm using that a lot as an example. It's a very famous pest in citrus production, and that's because it's drastically reshaped the citrus industry around the world. It spreads a disease called HLB. In Florida, where it is well established and has spread rapidly, it has cut citrus production in half, but still costs growers about 3 times as much now to operate their farms. In Ventura, we have ACP. It's ubiquitous, but the disease HLB has not spread beyond a few residential hotspots, which are in quarantine. And this pest has a natural enemy. It has a parasitoid wasp that's been released, but again, very little research on how habitat impacts this pest and parasitoid complex. So it's likely that habitat is important for pests in a couple of different ways. Citrus thrips, they're likely governed by landscape drivers because they're highly polyphagous. They feed on over 50 plant hosts, laurel sumac and berries and nuts and other crops. And for insects that have a lot of hosts, natural areas could be providing alternative food resources. Other pest species might use non-crop areas for overwintering habitat, or as means to hop around between different farms, basically. Habitat and landscape quality and composition could also influence parasitoids and predators. And again, that's because they need pollen and nectar to survive in addition to their prey insects. Here's the syrphid fly again. The adults need floral resources, even though the larvae are the ones who eat the pests. And they're very strong flyers, so they could be using natural areas or urban areas for food sources or habitat, or even to escape pesticide exposure. It's not necessarily that they're flying away to escape pesticides, but there could be a population of a beneficial insect on a farm and another population in an adjacent natural area. And if the farm population gets decimated by a spray, the natural area population could reseed the farm. The question that I have in this project is how habitat enhancements at local scales and at landscape scales influence these citrus pests and avocado pests and their enemies. Is one more important than the other, for example? And we honed in on farms using hedgerows specifically because that's a really popular form of habitat diversification in Ventura. Hedgerows are strips of perennial plants. They're usually drought tolerant, sometimes they're natives. Farmers grow them on field margins, and they've been found in several studies to create habitat and attract beneficial insects. They also can attract birds and other animals. There's challenges with them. They can require maintenance. They might not be effective unless there's a larger patch of habitat nearby that acts as a source of beneficial organisms. Many insects disperse and forage, though, over miles for food and nesting resources. And so, again, the landscape might be important. In Ventura specifically, we have the Santa Clara River Valley, which is a major conservation and restoration target, and it likely acts as habitat for many insects and other animals that frequent adjacent farmlands. So our experimental design for this study, which is ongoing, is aimed at assessing and comparing the impacts of large and small scale habitat patches. So we have lemon and avocado farms, and about half of them are close to the river, where there's like a lot of restoration programs right now. About half of them are distant from the river. And then about half of our sites have hedgerows on the margin, and half are managed for bare margins. And then we're also looking at riparian areas as well, and sampling in those zones to compare what organisms are found in less disturbed habitats. Okay, so our questions are, which are the features that are important for the insects? Is it local aspects of the farms like flowers and weeds? Is it landscape features like the percent of natural habitat or the amount of urbanization? And then we're comparing which are the features that are most important or most predictive of pests and beneficials and pest control. To look at insect pests and beneficials, we're using a few different methods. So we're using yellow sticky traps, which we leave on the trees for a week and put out every month for about 8 months out of the year. And then to assess pest pressure, we're utilizing PCA scouting reports. PCAs are pest control advisors, and that gives us a measurement of pest outbreaks that is kind of more similar to what growers use to make decisions. So we're using both of these methods because they provide us different information. The PCA reports are visual surveys. They're really great at seeing insects that don't move. And they also only capture insects that are important for growers. The sticky traps are biased towards flying insects, but they give us all the insects on the visit that fly, including those that, growers might overlook sometimes or might not care about as much. I have some results to share with you right now from some of our sticky traps. So this is what a sticky trap looks like. Each trap captures hundreds of insect specimens, which we identify to species when we can. We have, let's see, about 2 to 8 beneficial species on each trap, 6 to 11 species of pests. And so far, these are the most abundant insects that we have caught. We have found a lot of thrips, avocado thrips, and then citrus thrips. And then for natural enemies, the most abundant type that we have found has been parasitoids, which right now are all kind of lumped together into just like small parasitoids. We haven't identified them yet. That was an avocado. For lemons, it's the same. We have citrus thrips, we have avocado thrips, and then for natural enemies, mostly what we're finding is a lot of parasitoids. But what about the riparian habitat? I was kind of surprised to see what we found. I kind of had assumed we'd see pests and natural enemies that were good flyers like syrphids. But just like in the lemon and avocado farms, we found a ton of citrus thrips and avocado thrips, also some aphids. And then for natural enemies, again, we saw lots of parasitoids. Parasitoids are generally terrible flyers and disperse very passively. Another thing to note here is the scale of this graph. The total count is maxed out at 6K. On the previous graph, the scale went up to 3K. So we're not only seeing the same insects that we see in lemon and avocado farms, but we're just seeing like twice as much We're seeing a lot of them in riparian areas. So this kind of begs the question, what is the role of the riparian areas next to the farms? Is it attracting and hosting pests and predators that then move into the farms? Or is it keeping them in the riparian areas and away from the farms? Is it acting like a source or a sink of these insects? Still working on addressing this question, but we have some interesting preliminary data. So let's look at a breakdown of our sites by their distance to the riparian areas. I mean, this is a really crude categorization, but some of the sites were close and some were far to the riparian area. And we can kind of see a neat pattern emerging. So if you look at avocado first on the left, those sites that are close to riparian habitats have more natural enemies than sites that are far, and they have fewer pests. And then if you look at lemon, it's the exact same pattern. So sites close to riparian areas have more natural enemies and fewer pests. When we look at the landscape around each farm, we can categorize the percent of natural habitat around it, the amount of non-crop plants. So like basically any plant that's not lemon or avocado. This is like a more fine-tuned approach than broadly categorizing each farm as close or far to the river. This graph shows a relationship between non-crop habitat within 1 kilometer around each farm and the number of unique pest species. We're not seeing a huge relationship in the avocado farms, which is the green line. However, you see a kind of strong negative trend in lemons, where more habitat is associated with fewer pest species. And then if we do the same analysis, but with natural enemies, we see that as natural area increases, we see an increase in natural enemies more strongly in avocado, but both in avocado and in lemon. I wanted to look at thrips for a second because those are the most abundant pests that we've been catching at our sites. And I think comparing the two really illustrates why non-crop habitat doesn't always have the same effect on every pest. We know citrus thrips have dozens of hosts, and avocado thrips don't. Avocado thrips only have one host, avocado. We also know that natural enemies are not very effective at controlling avocado thrips, but they are at controlling citrus thrips, okay, or they can be, or it just has more natural enemies. So it maybe isn't super surprising to see that citrus thrips really respond to the amount of non-crop habitat, but avocado thrips don't really. And I think what this is really showing is that the effects of landscape, it's always going to be site dependent and crop dependent and pest dependent. So the results that I shared with you just now are really preliminary. We still have a lot more work that we're doing, and we have all that PCA survey data. So I'm really curious to see if we have impacts of natural habitat once we start analyzing that. So we'll be comparing the PCA survey data and our sticky trap data. We're still collecting pesticide use records for each site to see if habitat actually translates into lower pesticide use. We are also working with researchers who study birds, bats, and small mammals, and they've been collecting that data at the exact same sites. And so we can compare how habitat is impacting those organisms as well. Okay, I'm going to move on from biocontrol now, and I'm going to talk next about 2 projects related to chemical control. The first project is about resistance monitoring in avocado thrips. And then the second project, we'll be moving away from avocado. We'll talk about cabbage and diamondback moth. Okay, so just some background on thrips generally. I'm not sure how often you guys interface with thrips in urban IPM. I'd be interested to hear. I think I mentioned there are thousands of thrips species, and just about 1% of those are considered pests, including the avocado thrips. By the way, the singular for thrips is still thrips, so I'm not saying it incorrectly. It's always thrips. It's never thrip, which is kind of a weird thing. But 1% of the thrips species are pests. Some Some thrips species are what we call neutral, like they just feed on fungal spores or pollen or decaying vegetation. Some thrips are beneficial. They actually feed on other insects or mites and even other thrips. And even though these thrips have wings, they're poor flyers. They generally move long distances by floating with the wind, or they can be transported on infested plants or exotic species, and are periodically introduced. The major thrips that we have in avocado is the avocado thrips. And both the larvae and the adult can cause damage to the fruit. It causes a brown scabbing and scarring damage, sometimes called alligator skin. It can be confused with damage from wind or rubbing. And this damage, it doesn't harm the internal quality of the fruit, but it causes cosmetic damage. So it's a problem for our growers. It was first discovered in California in 1996. It was likely introduced from Mexico, and then it moved to Ventura, and then San Luis Obispo, and then San Diego. They've been a major pest with about 80% of our orchards reporting that they spray to control for thrips. And they really like coastal growing areas like Ventura because they're intolerant of high temperatures. There are a lot of thrips species that afflict our county, and they look very similar to some of the other thrips, but there's differences in coloration and behavior that can help you tell them apart. But basically, they have these fine, dark abdominal bands that transect the abdomen that help you identify them. They do have natural enemies, and the most important ones are the Franklinothrips, which have this almost wasp-like waist. They are mostly black. They might have these white or pale bands on the body, especially near the waist, and they're great predators, but it's unclear if they can breed quickly enough to control avocado thrips. There's some other predators as well that eat avocado thrips and other pests. Since we talked a little bit about conservation and biological control, I'll mention that a lot of these also rely on pollen and nectar. So lacewings eat pollen and nectar. Banded wing thrips supplement their diet with pollen and nectar. Yeah. The primary control product used for avocado thrips control is abamectin. It was registered in 2005 for avocado thrips, and it's a really good fit for avocado pest management because it has a very strong efficacy, even when it's applied on hillsides by helicopter, which is a big challenge for our growers. Most of them grow on the hills. It has persistent control, and it actually has a limited negative impact on natural enemies. But the problem with abamectin is resistance is very likely with it. It has a long persistence inside of leaf tissues, which means that sequential generations of thrips might be exposed to the same chemical product. And we know that chemical control is an issue that can lead to resistance. Generally, what resistance is when an insect population is subjected to an insecticide, it kills the most susceptible individuals and leaves behind those that are less susceptible, and those reproduce and increase the population. So in other words, the pesticide is no longer effectively killing a substantial number of individuals in the pest population. And we're concerned because there's hundreds of species that have developed resistance to pesticides in the last 50 years. And it's really hard to get new pesticide products on the market due to regulatory and environmental issues. Insecticide development is typically very slow, and resistance will outpace the discovery and registration of new chemistries and products. Let's kind of dive into resistance a little bit more and how it plays out over multiple generations. If this is your pest population, let's say you now apply a pesticide. The gray are dead insects. These were vulnerable individuals, and the orange are the ones who survived. They have some sort of resistance to the product. You can see that most of the insects did die, but then you have these orange insects. They have an inheritable trait that allows them to live, that they can pass on. And some time passes, maybe some new pests arrive, and the old ones are around and mating too. They have some offspring, and they passed on the genetic basis for that trait. So you spray them again, and maybe you knock off some of them, but this time more survive because there's more insects with those survival traits in the population, and they keep reproducing. And every time you spray, you see that there's more and more of the resistant insects left over. You're actually putting selection pressure on the population every time you spray. You're killing the vulnerable population and leaving resistant individuals behind, who can then mate and go on. And what you end up with is a population that displays resistance to a pesticide. And it actually indicates that there was a change in the genetic makeup of that insect population that occurred over that time. Individuals within this resistant population are known as a biotype. They have unique biological traits. Okay, so that's not common to the population as a whole. Thrips very commonly develop resistance. They lay a lot of eggs in a short amount of time, and then they also have asexual reproduction, which means that a female can lay eggs and reproduce without mating. So if she has a trait that allows her to evade a pesticide, then that trait won't be diluted in a mating event. It just gets passed on. So it's basically a numbers game with thrips. And there are over 150 cases of resistance associated with thrips, not necessarily with avocado thrips, but with other species. I wanted to talk about the mechanisms of resistance and how it evolves, because I think it's kind of interesting and I didn't know about it until recently. But the most common resistance mechanism that occurs in insects is called metabolic resistance. That's basically when insects use enzymes to break down and detox pesticides, and then they secrete the broken down products as waste or excrement. We see this a lot in polyphagous insects, so insects that eat a lot of different plant species. Those insects have evolved the ability to eat a lot of different plants, and so they have genes that code for enzyme systems that can break down and convert different chemicals. They basically convert hydrophobic compounds to less biologically active hydrophilic compounds, that can be metabolized and excreted out as waste. So just to summarize, insects that are highly polyphagous have metabolic detox abilities. And so Western flower thrips are insects like that with a diverse diet, and we see that they have metabolic detox abilities. The second most common form of resistance is physiological resistance. And in this type of resistance, the insect evolved a modification to the site where the active ingredient of the pesticide binds to the insect. So for abamectin with avocado thrips, we know that it binds to what's called a glutamate-gated chloride channel. It's an ion channel in the membrane of the nerve and muscle cells. And when abamectin binds to that channel, it changes the ability of the membrane to control the intake of chloride ions. And as a result, the cells get flooded out with ions, and then the insect dies. So if an insect were to show physiological resistance to abamectin, you would see that they would evolve a modified chloride channel so that the pesticide wouldn't bind to it. Okay, penetration resistance is what happens when an insect's outer cuticle develops barriers, which slow the absorption of the chemical into its body. This protects insects from a wide range of insecticides. The kind of most common example of this is bed bugs. That's why chemical attacks are so difficult with bed bugs. They have a really thick cuticle. And then lastly, there's behavioral resistance. Insects may detect or recognize a danger and avoid a toxin. We've seen this mechanism of resistance been reported for several classes of insecticides, including organophosphates and carbamates and pyrethroids. Insects might stop feeding if they come across a certain insecticide, or they might leave an area, or they might move to the underside of a sprayed leaf. Thrips are naturally very cryptic. They naturally prefer to reside in enclosed, concealed spaces, and that's thought to reduce their exposure to insecticides. And those traits might be something that are actually passed on, traits for kind of that cryptic hiding behavior. So there's a lot of factors that can cause pesticide resistance. Basically, any factor that weakens the effectiveness of a pesticide can favor the development of resistance. So improper pesticide selection, using an incorrect pesticide dose, poor timing, an unfavorable environmental condition, or a low quality pesticide, maybe that one that's been degraded. If your pesticide is old or been exposed to the sun and humidity, it might be less effective. pH is also important. Sometimes spray solutions should not go over a pH of 7 or they start to break down. And then how quickly insecticide resistance develops also depends on other factors like the type of insecticide used, how often it's used, and then on the insect and how capable it is at detoxing that insecticide. So there's a lot of factors at play here. There's some things we can do to prolong insecticide efficacy for avocado thrips or for any other pest. Generally, we advise growers to limit the frequency of applications by spraying only when necessary, and then to alternate between insecticides with different modes of actions. So when I say mode of action, that's how a pesticide impacts an insect. And we rotate modes of actions because we assume that the frequency of resistant individuals to one insecticide will be knocked down during the application of another insecticide with a different mode of action. You can find the mode of action on the label, but there's also a number designation for that mode of action on what's called the IRAC website. IRAC is the Insecticide Resistance Action Committee, and they have an app, and you can look at the mode of action and learn more about the chemical compounds in a pesticide's active ingredient. It really helps you avoid using products back to back with the same mode of action. This is a table of miticides. And I think it illustrates why it's important to rotate modes of action and not just product names. I got this from Raymond Cloyd at Kansas State. It's products that are used for mite control in greenhouses. And what the table shows are the active ingredients of different products, the trade names. Then there's activity type, whether it's contact or translaminar or systemic. And what I want to highlight is that there are 14 products here, but there's only 6 modes of action. Each mode of action is highlighted with the same color if it's the same mode of action. So for example, there are 6 products labeled white because they are mitochondrion electron transport inhibitors. And so it seems like there's a great diversity of products available for mite control in greenhouses, but it's actually less. Another strategy for reducing resistance, I've never seen any grower do this, but I'd be really interested to see if any are willing in the future, is to leave areas of an orchard untreated. This does a couple of things. So it could conserve natural enemies that can then spill over into the other parts of the farm after a spray. And then it also allows for the survival of susceptible pests that can breed with pests that have resistance genes. So it reduces the rate at which resistance develops and resistance traits are passed on through the whole population. And then finally, another thing we can do is monitor populations for changes in susceptibility to insecticides. And I think this is really important for avocado thrips. We have really limited control options for avocado thrips. We basically have one product that every grower uses, and it will not be easily replaced if it's lost to resistance. And abamectin, it's contact, it's translaminar, it kills juveniles and adults. Sometimes it's used twice a year by growers, and no one has been tracking resistance to it. So my colleague and I at UC Riverside, we just found out 2 weeks ago that we obtained some research funding to start doing this. So we're really excited. We're going to be developing a resistance monitoring program for avocado thrips. We're going to start off at a small set of farms across the region, and we're going to collect thrips. We're going to rear them in the lab on leaves that have been dipped in a pesticide solution, and then see how effective those pesticides are at killing the thrips. And then hopefully we'll expand out our testing to become a diagnostic service for growers in the future. Once we get the data, our goal is to create a map or another tool to share online with our community so our growers can start looking for resistance hotspots in our county. So we're really excited about this project. I don't have any data to share yet with you though, but maybe next year or the year after. Okay, now I have one last project to talk about in terms of chemical control. I know I'm throwing a lot of different projects at you that are all very different, but I wanted to give you a taste of different things that we do here. This project is about diamondback moth and cabbage. So diamondback moth, or DBM, probably originated in Europe, but now it's found throughout the world. It's basically found wherever brassicas are grown, and it is a pest of cruciferous plants. So it attacks broccoli, cauliflower, cabbage, Brussels sprouts, kale, etc. And it's incredibly threatening. The caterpillar chew on the plant and they leave these big holes and they make the plant totally unmarketable. It's very hard to control for a number of reasons. It doesn't have effective natural enemies. We know that the moth migrates long distances and it does have parasitoids, but those tend to lack the same flight capabilities to follow it around. It's very good at evading enemies. They wriggle vigorously when they're threatened or stimulated. There was a study showed that they can actually wriggle backwards and hang off leaves when confronted by ants. And moving backwards is a very hard skill for insects. That's not commonly seen. Even a lot of animals can't move backwards. So that's a pretty cool thing that it does. And then it's also really hard to control because of pesticide resistance. It has a long history of becoming resistant to insecticide, starting with DDT in the 50s. Since then, it's become resistant to basically every new insecticide that's arrived on the market to control it. In fact, sometimes this insect is used as a genetic model for studying resistance because resistance genes just evolve really quickly in populations of DBM. Okay, so Syngenta is planning on releasing a new product for DBM. It's called plinazolin. And I ran a small trial this past summer with some of my colleagues to look at the efficacy of this new product and see is it something our growers can use in the face of diamondback moth. So we had 3 treatments. We had beds of cabbage spaced by empty buffer beds, and each bed received either the new product, plinazolin, or a conventional standard, like something that's commonly used in our area. So we used Radiant, or it received no treatment. And then we replicated our plot 4 times. And each of the plots, the pesticide treatment was randomly assigned to each bed. And then after we applied pesticides, we came back, we counted diamondback moth larvae at multiple intervals after application. So at 2, 7, 14, and 21 days after application. And we would go through and select plants randomly and count the larvae. I'm going to show you the results in numbers, but I won't make you wait for the takeaway point. In this case, I think a picture is worth 1,000 words and represent our results pretty well. So the plinazolin treated plants, they looked fantastic. They were super clean. Our control and conventional standards did pretty poorly. For the most part, Radiant performed better than our control, but not always. Assuming our local population has resistance to Radiant, at least partially. This is the time series that shows the average number of moth larvae per plant and how that changed across the experiment. The red line is the plinazolin treated plants, and then the yellow is the Radiant, and the blue is the control. And then the little up and down lines are standard error, which show how much variation in diamondback moth larvae the plants had at that date. So in this graph at the bottom, 0 is the pesticide application date. You can see that the plinazolin treated plants, which are in red, have much less moth pressure throughout the study. And then applying Radiant is marginally better than doing nothing. This graph is basically another way of showing the same thing. It's the same data. And again, you can see that the red, the plinazolin treated plants have lower numbers of the moth. And when we use statistical tests, we found that these differences at every sampling date were significant. Plinazolin suppressed the pest from 67 to 80% control compared to our untreated plants. And then we looked at yield at the end of the study and saw that, wow, the plinazolin had greater yield. There were more heads that we could pull out. So our growers are really happy about this. We also, at a subset of the sampling dates, just for fun, kind of counted parasitized larvae. So that's larvae that shows that they'd been attacked by parasitoid wasps. We did see a lot fewer parasitized moths in the planozolan treatments. We weren't sure if planozolan was repellent to natural enemies or if there just weren't enough larvae in those beds to be attractive to the wasps. So it doesn't really say anything about the impact of planozolan on the natural enemies, but it's an important thing to think about is how these really new effective products impact beneficial insects. The takeaway point from this trial is that current insecticide use is really heavy in cabbage and it's not working well. My colleague Oleg previously tested a lot of different products for DBM and they found that none of them were significantly better than just using water to wash off the plants. And so having this new product is going to be really important for managing this pest and hopefully reduces pesticide use overall in the crop. But we also have to conserve this product. Oh, was that a question or no? I don't think so. Okay.
Mostly paint.
Okay. So with resistance being such a big issue for diamondback moth, I wanted just for this last part of the talk today, discuss an alternative form to moth control called mating disruption. And it takes advantage of the way that moths mate. Male moths find female moths by following their pheromone scents. And they basically they fly around until they find a plume of pheromone. And as they follow the plume, if they get to the edge of the plume, they start to turn around so that they stay in the plume. And they keep turning around so they stay within the plume and find the female. With mating disruption, you have a technique that releases synthetic sex pheromones and it confuses the males. Basically, if there's a lot of pheromone in the air, they can't effectively hone in on those females and mate with them. Mating disruption is used widely in apples and some other crops. For brassicas, there is a product currently available, but most growers don't use it. And there's other products that are in development and about to hit the market soon. So we're trying to figure out which of these products is most effective and how are they most effective? Do they need to be applied at a certain scale across a certain amount of acreage? We want to know why growers aren't using it. Is it the cost or is it too difficult to implement for other reasons? This is a project that's just starting up this year, so I don't have any data yet from it either. It's led by Dr. Ian Grettenberger at UC Davis, and the team includes researchers from CDFA and USDA as well. We have 4 main objectives. First, we're going to be focusing on evaluating products and practices around mating disruption. And then another part of this project is learning more about how diamondback moth disperse and utilize natural areas and non-crop hosts. We're going to be asking about their interactions with parasitoids, and then lastly, we'll be tracking their resistance levels. So for the non-crop host part of this project, we know diamondback moth exploits a number of non-crop hosts, basically any brassica it can find. In Ventura, weedy brassicas are abundant in the landscape. Even right now, if I just look outside the window at the hillside, I can see the hills are covered in mustard. So it's kind of coming to an end right now. And as it comes to an end and dies out, all the diamondback moth are going to come down from the hillside and go into our farms. When I think about mustards in the hillsides, on one hand, it could be a bad thing if mustards are an alternative host and habitat resource, but it might be a good thing because it could be a refuge where the moth evade pesticide sprays and stay susceptible to pesticide products. Those susceptible moths can then mate with in-field resistant populations and prevent pesticide resistance. So we need to figure out which species exactly in the non-crop areas are harboring diamondback moth, what time of year are they harboring them, and then if the populations in those non-crop areas have different resistance levels than in neighboring farms. So that's part of this project. For parasitism, we're going to be collecting larvae across different regions of the state, and we just want to develop a basic understanding of parasitism rates for the pest. How ubiquitous is the parasitoid, and is that parasitoid also using non-crop areas? And then at various points in the past, there's been efforts to track pesticide resistance for diamondback moth, and those studies have found that Ventura is a big hotspot for resistance. So we're going to continue monitoring resistance across production regions in California for about 4 years of this study for different key materials with different modes of action.